Hi everyone, I'm the Plant Propagator and welcome to my channel. If you're interested in learning a little bit more about the work that I do, please subscribe to my channel. Today we're in the laboratory and I want to talk a little bit of science with you and we'll see how it goes. Don't turn it off, don't turn me off. I've got a little experiment that I want to go over with you and tell you a little bit more about the type of work that I'm doing and the type of science that leads us forward. So what I want to talk to you about is some media addenda to the orchid media that I'm using in my laboratory. The basic media I developed on my own. It's very similar to what many other people are using, and I made some slight changes to it. I also, just so you know, I've been working with the plant tissue culture, and I've been working in the laboratory for over 40 years. But I've only been working with orchids in the laboratory seriously for the past couple of years, and I'm learning a lot as I go. So the first experiments that I did not work out very well. I put seeds in culture. I tried growing the seeds, and they did okay, but not real well. I didn't get very good survival of the orchids when I took them out of culture and put them in bark. And recently I've had some really nice success, very, very high efficiency recovery, and that was because of a medium addenda, something that chemical or a compound or a substance that somebody told me to add to the medium that would give good results. So I was working with some colleagues, and they suggested that I add Beech-Nut baby food formula, stage 2 banana. So I hate whenever somebody tells me this. I just really don't like doing it because this is undefined. You don't know what's in there. In the early days of plant tissue culture, people used to add coconut milk, for example, to the medium, but as we moved along, figured out that the coconut milk contains some special vitamins and some hormones. And instead of putting coconut milk, which can vary from nut to nut, it's better to add chemicals, the stuff that's up on the shelves in here, and those are much more defined. So I like using those. When the colleague told me to use the Beech-Nut baby food formula banana stage 2, I did that. But I did add it, and I did a plus and minus the formula. And what I found out, and I did an experiment, and I collected images, what I found out is that it did work. It helped out tremendously. I got incredible root growth on the replate medium, and that is when the plants are allowed to develop and grow roots in flasks. And it was clear. And I tried 3 different orchids, and it worked. And so I did that again, and what that means is that, okay, I need to figure out what's going on here and what's in this formula. So what I was told to use, this is what you use. This is Beech-Nut banana phase 2, or I'm sorry, stage 2. And what's in here, you look at what's in here, and I'm going to get really close, but what's in here is not much. Banana, water, and lemon juice concentrate. And so that's what's in here. One thing I should mention is that when you add this to the medium, what happens is the pH gets lowered a lot, which means that there is a fair amount of lemon juice concentrate in there, and that the lemon juice contains various things that really lower the pH. So I still didn't know what was going on, and I'm not CSI in my laboratory here. I can't analyze it. So I contacted the people at Beech-Nut, and they were very, very helpful. And what they told me is that this baby food contains 0.5% lemon juice concentrate, which really helped me out a lot. And when you do that type of analysis, you can determine how much of the components of lemon juice are actually in there. You go to the lemon juice concentrate websites and you look at that, and it's standard practice to add 400 grams per liter to have, I'm sorry, not add, but to have 400 grams per liter of citric acid, or 400 milligrams per milliliter. Don't turn me off, this is not hard, in the lemon juice concentrate. So if you do the calculations, what that ends up with is 225 to 230 milligrams of citric acid is what I add to a liter of medium. And that's the major component. And the reason that citric acid caught my eye is because it is actually commonly added to plant tissue culture medium. It is it's a reducing compound, and what it does, what it probably does in here is it keeps the bananas from turning brown. So this is a banana, and it's starting to turn brown, and if you would put this in a blender and just let it sit, it'll turn brown. So the lemon juice keeps the banana and the formula from not looking too nasty and turning brown. But it's also can be used in plant tissue culture to prevent browning of some of the plant cells and plant tissues in culture. So it kind of makes sense that the component that's in here that may be contributing is the citric acid. So I'm going to do an experiment where I'm going to add, and when you do experiments, you have to do plus minus. A lot of people that you see say, I did the science, I did research, I looked it up online. That's only a really small bit of science and what you should be doing when you do a proper scientific experiment. So I have to try a series of media that I have plus and minus the citric acid to see what happens. I have to try adding banana, grind up banana, and then I'll add as another control, because this works and we know it works, is I'll add the Beech-Nut baby food formula. And what I'll hopefully be able to determine whether it's the banana, the citric acid, or the baby food formula, and if it's not the citric acid or the banana, then it's too bad. I'm not going to be able to figure out what's going on. Even with a banana, it's made up of a number of different components. My hypothesis going in is that the citric acid as a reducing agent is what's contributing to this really fast growth. But we'll see, and that's why we do science. All right, and so again, we're going to do some proper science, and we're not just going to make stuff up or look online and come up with a hypothesis and not evaluate it. So we'll do evaluations. Again, what you see, these are some of the flasks that contain the baby food formula. And you can see with this dendrobium here, that there's really extensive root growth. Even with a larger container shown right here, you can take a look at the really incredible amount of root growth. So this is a dendrobium. You see the same thing with this is a Laelia pumila, which is a Cattleya type, but look at the incredible root growth. And what happens actually, and this is a Cattleya, this is again but look at those monster roots. And what you find out with these things, when you have orchids in flasks, what I'm seeing is that the survival rate is just really high if these things have nice root systems. So I'm going to do a little bit more of medium optimization and try to discover what actually is in the baby food formula that contributes to this beautiful growth that we have in these flasks containing these orchids. Again, different orchid types here. It looks like it contributes to things. I have a number of orchid seedlings that are in the laboratory that as soon as they're a little bit larger, I'll make the media and transfer them to the various media that have the various components, and we'll try to discover together what contributes. Well, first of all, we'll see if I can repeat the work and decide and try to determine what contributes to this really nice root growth. So that's all I have for today. I hope you didn't turn me off during this lab exercise, and happy propagating.